This is a tutorial for the new qPCR feature of KCET 7. That stands for quantitative PCR. This shows the contents of the KCET version 7 PC folder. Now I'm going to run this on a Mac. You can run it on either Windows or the Mac operating system. I will double click on the executable file, which will take a few seconds to open. So this is the opening screen for the KCET simulation. Click here to begin, and then click to continue. You come to the data screen. On the silver button bar of the data screen is a new button, the qPCR button. Before I click that, I'm going to close these windows since we don't really need them. And I'll use the qPCR button to open a file containing qPCR fluorescence chart data. This is actual data from a qPCR machine. The data set that I'll use is actual data from a honeybee experiment. And it's called qPCR honeybee. So I will highlight that and open it, reposition these windows that appear. Notice there is a line, click the load button below. So that refers to this small button, the load button. And 96 wells appear that are loaded, and you can tell that they're loaded because they are orange in color. Now some of them, a few of them in the lower right hand corner are blanks, but the rest of them have actual data from a honeybee experiment for 10 different hives, a reference gene, actin, and then samples that might include deformed wing virus, which is what DWV stands for, or black queen cell virus, which is what BQCV stands for. Again, that's deformed wing virus and black queen cell virus. So the point of this is to try to see what are the relative changes in these viral levels in the 10 hives. That's our overall goal. And this is a use of relative qPCR as opposed to absolute quantities of DNA, which we'll demonstrate later when we talk about standard curves. So the first thing we want to do is run this experiment for all 96 wells. And to do that, I'll use the qPCR button. In fact, I'm going to bring the graph forward before I do that. I'll use the qPCR button, run qPCR for orange wells only. They're all orange, so this would be an appropriate command. And you'll notice as that data is run, curves appear that are associated with what are called CT values. And you can see that for all of these, except for the blanks in the lower right-hand corner, there is a CT value associated with almost all of these wells. And the question is, what does that mean? Well, to make that understandable, we need to have fewer than 96 wells. And I'm just going to select one well. For one, this will be well A3 for hive 1 actin. Notice the CT value is 26.09. We can rerun the qPCR just for that one well by clicking the qPCR button, run qPCR for purple wells only, and it will show just that one well. Now if you look carefully, these are cycles in the qPCR machine from 0 to 45. So DNA would be doubling each cycle, which is a little hard to tell because it takes a while to get going. But once it reaches a certain point, you reach a phase of exponential growth of DNA, of the copies, and eventually the number is going to cross a predetermined threshold, which we've arbitrarily set as 4051 in this case. This is a movable threshold. I won't move it right now. But that is a fluorescence value because the fluorescence values are on the y-axis, the vertical axis, and then the cycle values are on the x-axis, the horizontal axis. So the CT value of 26.09, if you hold the mouse cursor over the point where that line crosses the threshold, you get 2609. So that corresponds to this value right here. To make that a little easier to see, you can magnify the graph by clicking this icon with the magnifying glass and then dragging this up. And then I will position the mouse cursor over the intersection point. Whoops, I just moved it a little bit. I'm going to put it back. 4051. And there is 26.09. Now this is going to change a little bit depending on which pixel you hit. So it's not going to be exactly the same, but it'll be very, very close to what you see in those labels. I'm going to click this magnifying icon again. It makes the chart much smaller, a little too small, so I'm going to click it another time and bring it back to its original size. So that is what a CT value is. It's the point at which a curve crosses the threshold. In reality, these are done in triplicate. So I'm going to highlight all three of those, and I'll rerun qPCR for purple wells only. And you can see that those three are close together, and you would hopefully those would be close together. They wouldn't wildly diverge, otherwise you might have a problem with your experimental setup. Now, one thing we would like to see is an average for those three values, and we can calculate that a little bit later. Let's compare the actin with the deformed wing virus or DWV virus samples. And to do that, I can highlight all 6 of those, rerun the qPCR for purple wells only. You'll see those values are all pretty close together. You can tell visually from the graph, and you can also see it from the range here, which is all pretty close to 26 for a CT value. So it just kind of an informal look at this would say there's really not much of a difference between the reference DNA, which is actin, and the deformed wing virus DNA. I'm going to click on these 3 wells to deselect, to get them back to their original state, make them orange. I'm going to drag to select well 7, 8, 9, and right now it says no CT because we hadn't selected them before. But I'm going to rerun this with purple wells only and see what happens. So now we actually do have CT values for the 3 actin reference samples and for the BQCV samples. Now notice, just visually looking at these 26 versus 34, and you can see the difference here. So I'll just kind of hold in this particular region just to get an idea for these wells. I can get that to work, 32.5. So roughly 33 for those 3, and roughly in this area somewhere in the vicinity of 26. So what you would like to do is get the average value of these 3, the average value of these 3, and subtract those 2 values to get what's called a delta CT value, delta standing for change. And we can do that with the KCIT software by clicking on this delta CT button. When you do this, you get a value of minus 7.49, which is the relative change between the average for the 3 actin and the average for the 3 BQCV samples. That is depicted visually in bar chart form. So this is 0, and this would go down minus 0.749 units. Now the reason why it goes down instead of up has to do with how qPCR works. The sooner a curve hits the threshold, the more DNA you have. If you have a greater starting quantity, you're going to end up hitting the threshold quicker than if you have less of a starting quantity. So this represents more starting DNA than this. However, the CT value here is less than the CT value here. Again, smaller CT values are associated with more initial copies of DNA. The way this works by convention is we're going to subtract the average value of this larger number from the average value of this smaller number, so you end up with a negative value, and that's why the bar goes down. You might have to think about that a little bit, but that's the convention that's used with qPCR. What we're really interested in, though, are the delta CT values for the entire data set. So there's two ways of doing that. I could either reopen the file. If I reopen the file, everything will be orange, or since I've only got 6 purple values here, it's just as easy for me to slowly click on these 3 wells. You do have to click rather slowly for this to work right. And now I'm going to rerun qPCR for the orange wells only. And click Delta CT, this button. So now we get the complete set of red and blue bars. Blue is for deformed wing virus. Red is for black queen cell virus. Even more interesting is a delta-delta CT comparison. In other words, comparing 2 delta CT values. For example, Hive 1 BQCV of minus 7.49 with Hive 5 BQCV of minus 5.72. Hive 1 is the same as sample 1, so minus 7.49 would be this red bar, and minus 5.72 would be this red bar. To determine that, you click, I'm going to single click on the red bar, and that puts minus 7.49 in the numerator of the fold change formula. It's 2 raised to that difference, minus 7.49. And I will double click on the red bar under sample 5 or Hive 5. So that puts the minus 5.72 in the denominator, 2 raised to that power. If you do that calculation, the value is 0.29 fold change. You could do the same thing with comparing Hive 1 with Hive 10. I'll double click on that, and that's a 17.88 fold change. That is how you determine CT values, delta CT values, and delta-delta CT values using the K-SIT simulation. And this would work with any set of fluorescent data. It does have to be in the proper format for K-SIT to work. If you look at the actual file, you'll see what that format is. So if you have your own fluorescence data, this simulation will display it. It's very similar to how the software displays this information that comes with the qPCR machine. Next, we want to show how the K-SIT simulation can be used to generate a standard curve that could be used to come up with an absolute estimate of the number of copies of DNA present in a sample. So that's different from what we did before with the delta CT, delta-delta CT procedure. You're dealing with relative changes for those particular kinds of experiments. But for absolute estimates of DNA present, you need to run a standard curve. To demonstrate that, after using the clear button to clear everything, use the qPCR button and open a file, and this time we're going to open the file that says standard curve qPCR data with an outlier. And I'll explain why the outlier is present in just a minute. So as before, we need to click the load button every time you open one of these files. And in this case, only 14 wells are loaded. You'll notice there is a replicate for each one of these. And if you look at the labels, the label indicates the initial copies of DNA present, the known initial copies present. So this is set up deliberately so we know the DNA so that we can generate a curve that can be used for predictive purposes. This is E notation. This stands for 1 times 10 to the first power, or 10. 1 times 10 raised to the second power, or 100 copies. 1e plus 3 is 1 times 10 to the third power, or 1,000 copies. 10,000, 100,000, a million, and 10 million copies. So those are known quantities in the samples. And notice there is a replicate for each one of these. And usually you have 3 values, but in this case we only have 2. To run the standard curve, we'll go back to the qPCR button, run qPCR for the orange wells only. And you'll notice something is a little odd here, because you can see the pairs, which you would assume that those replicates should be pretty close in terms of their CT values. If I move this over, you can see that better. So again, the CT value is where a curve is going to meet the threshold. So here are the CT values: 10, 10, 13, 13, 16, 16, 19, 34. Now that looks suspicious. 23, 23, 26, 27, 30. The rest of them look fairly reasonable, but this one looks way off. In fact, if you go over to the curve itself and hold the mouse cursor over it, you'll see that's well D2, and well D2 is right here, this one, and that is the outlier. So to see what that would look like on a standard curve, we click the standard curve button. And a standard curve is basically plotting the data points, the CT values versus the initial copies. So the top line would be the initial copies. The second line for each one of these cells is the CT value. So it's plotting those. There's a negative relationship between them. And then you use a regression analysis to plot the line that best fits the data. Well, this line's a little bit off because of the presence of the outlier. And you can tell that because there's an R-squared value of 0.774. If those data points fell exactly on the line, the R-squared value would be equal to 1.0 or very close to it. Here is the formula for the regression line, and I'll talk about this in a minute. First thing we want to do, though, is to eliminate this outlier. And normally you have to be very careful about outliers. Sometimes outliers might be your most important data, but in this case it's a standard curve and the lines should go through those data points, so we know this data point is wrong. Something was wrong with the experimental procedure. And before we rerun it, though, we need to go back to the amplification plot. This is just a thumbnail of that amplification plot. You need to click this button to go back, and then go to the qPCR button and run qPCR for orange wells only, and that will exclude the purple well. And if you keep your eye on that purple curve on the far right, you'll notice that disappears because it's not included in this particular analysis. We go back to the standard curve, and as before, we've got an R-squared value, but this time it's 0.999, which is very close to 1, and that's because these data points fall almost exactly on top of that regression line. Now you'll notice here it says 15 copies. The actual default that the program has is 20. I'm going to change that back to 20 for right now, because this is what you'll see if you run the program from scratch. So for a CT value of 20, the interpolated copies are 1.056 times 10 raised to the fourth power. You can see that from the line itself if you hold the cursor in the general vicinity of 20. Now that's 1.095 times 10 to the fourth, which is a little high. Here's 1.06 times 10 to the fourth, which is closer. So it's not going to be exactly the same because of the way the program works and because it's actually the cursor's over a particular pixel on the screen, which throws things off just a little bit. To make it a little easier to see, we can magnify it and try that again, holding it on one of those black pixels that lines up pretty close to 20. And there's 1.026, which is pretty close to 1.056. So it just shows that if you had a CT value of 20 for some unknown sample, and you use this regression line to predict the number of copies that you would predict are somewhere in the vicinity of 1.03 times 10 to the fourth. copies of DNA present. So it's a way of estimating the number of DNA copies in an unknown sample. To do that, you've got to set up a line like this based on known quantities of DNA. I'm going to go back to the magnifying icon and make it the size it was before. And just before we leave, I'm going to change that to maybe a number like 10. As I do that, the number of interpolated copies is going to change as well. Here's another example. If we held it on this location, that would correspond to a CT of a little over 8 and an interpolated copies of 3.38 times 10 to the 7th power. So this concludes the tutorial on qPCR.